firstly, we're going to be just do a general overview on muscles as a whole. So what are the different muscle types? So there are 3 types in our bodies. There's skeletal, cardiac, and smooth. Each type is used in different systems and for different purposes. Like I mentioned, today we'll be mainly focusing on skeletal, as the other 2 will come up in other lectures. But I want to show you what the difference is between all 3. So all the way on the left, you have smooth muscle. The reason it's called smooth muscle is because it's the only muscle type of the 3 that's not striated. You can see right here, not striated. So when you look at it, it actually looks smooth. Here's like a picture of it under a microscope. Whereas if you look at these 2 types of muscles, they have these little like lines over it. Those are called striations, and that's why they're referred to as striated. Whereas smooth muscle is not. Smooth muscle mainly is found in walls of internal organs, as you can see here, in your GI tract, in your kidneys, in your liver, everywhere, most of your organs, the walls, and even in your blood vessels, the walls have smooth muscle. Okay. The muscles are controlled involuntarily, just like you don't voluntarily control whether or not your stomach expands or contracts, or whether your blood vessels contract or expand voluntarily. Your body does that automatically for you, right? Next is cardiac muscle, and as the name suggests, this is muscle that only is found in the heart, right? It's unlike in smooth muscle, it's striated as well, right? So it has these little lines on it. And just like smooth muscle, it's involuntary, meaning that you don't control how fast your heart beats or contracts. Your body does that automatically for you. Lastly, skeletal muscle, which is what we're going to be talking about mostly today. It's striated. The fibers are striated, meaning, again, they have these lines, these striations. It's usually attached to your skeleton. So, for example, if you can see here, this looks like a leg, right? The muscles are actually attached to the bones of the leg, right? And they pull on the bones. That's why it's called skeletal muscle. And the 2 unique things about skeletal muscle is, first, unlike the other types, it's voluntary. You choose when you want to like move your arm or move your leg or move your head or anything like that. You choose how you can voluntarily move these muscles at any time. And the other difference is that the muscle fibers in skeletal muscle are multinucleated, meaning if you can see here, there's these muscle fibers, they have multiple nucleuses per fiber, whereas the other 2 types, if you can see here, are uninucleated, meaning they only have 1 nucleus per fiber, okay? I would recommend actually memorizing this like picture or the information that's present in this picture, mainly each type of muscle, whether it's striated or not striated, whether it's voluntary or involuntary, where each muscle is found. So smooth muscle in organs, cardiac muscle in the heart, and skeletal muscle attached to skeleton. Those are the main things I would focus on, right? In terms of differentiating between the different types of muscle. A lot of different things for muscle contraction are actually fairly similar for all types of muscle. So you would be able to like transfer some knowledge between, okay? But let's move on to skeletal muscle. So this is what the diagram for skeletal muscle looks like. So as you can see, this is a muscle fiber, right? It's this long tube, and there are the striations here with the lines. Sorry about that. And as I mentioned, multinucleated. So you can see there's nucleuses everywhere here, right? And obviously mitochondria all around to provide energy. The sarcolemma is kind of like the membrane, right? The membrane for a muscle fiber. And there's these things called myofibrils, these little smaller tubes inside this big tube, right? And we can actually see a close-up of it right here. Here we can also see the sarcoplasmic reticulum, which is very similar to the endoplasmic reticulum in any other cell, except in muscle cells, it's called the sarcoplasmic reticulum. If you notice, everything in muscle cells is just called sarco something, sarcoplasmic reticulum, sarcolemma, right? That just signifies that it's part of the muscle. So you notice there's in a myofibril, and this is something that we'll be covering a lot, there's this thing called a sarcomere, right? If you notice there's this Z disc right here, this green line, and then a Z disc on this side, and there's many of these on a myofibril and many more in a muscle fiber. The length between one Z disc to another Z disc is called a sarcomere. It's a unit of measurement, and in making up a sarcomere are different types of filaments. So you can see here, these dark purple ones are called thick filaments or myosin, and these thin ones are called thin filaments or actin. And as well as there's these, we break up this big unit, the sarcomere, into smaller units called the I band. Sorry, my mouse. The I band, the A band, the H zone. Over here, there's this M line, and actually it's right here as well. But this is for a different sarcomere. So if you had another Z band right here, this would be the M line for that sarcomere. But for this sarcomere, the M line is this these three lines here. It's the midpoint, right? So as shown in the previous slide, a sarcomere is a unit of a myofibril from Z disc to Z disc. It's how a muscle contraction occurs. During muscle contraction, sarcomere length decreases. So if you want to think of this like sarcomere, when muscles contract, the myofibril contracts, and actually specifically the sarcomere itself gets shorter, right? Gets contracted. There are four components of a sarcomere that you should know: the I band, the A band, the H zone, and the M line, as well as the Z line, right? A quick way I memorized this is, you know that in order it goes I, A, H, M, and then from both sides, so it would be H, A, I, right? This is what like a sarcomere, but really you just have to know IHM, right? I am, and that's a good way to memorize like from left to right or from right to left, going from the outside to the inside, the direction, the order of the different bands or zones. Okay, I am. Okay. Now, again, I'm showing a picture of a sarcomere, right? These are the Z lines at the edges, the Z discs. The M line here in the middle is not shown, but this big red thing is the myosin, the thick filament, and these little blue lines are the thin filament, and these are the zones, okay? So in the I band, it's only thin filament. So if you see here, the I band ends right here. There's only these blue lines. There's none of these red thick filaments. In the A band, which is next, there's both these blue lines and these red thick filaments. In the H zone, there's only these red thick filaments. There's none of these blue lines, okay? An important note is that during contraction, only the thin filament moves and actually contracts. Thick filament doesn't. This means that while during contraction, sarcomere length, H zone length, and I band length decreases, A band length doesn't. So let's explain that. If you notice, an A band is from here to here. This is actually the whole length of the thick filament, right? So if you see this red line, the A band is always the length of the thick filament. If the thick filament never gets shorter because it doesn't contract, the A band doesn't get shorter during muscle contraction, right? Whereas the thin filament does. So if you want to think of it, the thin filament, the blue, is going this way and this way. Eventually, all of this H zone, if this moves closer to here and this moves closer to here, the H zone will only be this small part that hasn't been reached yet, this new part. The H zone will have gone from this size to this size, right? So it would have shrunk. Similarly, with the A band is only where the thin filament is. Well, if the thin filament and the walls, these black bars, the Z discs, move closer to here, well, now the A band is only this length rather than this length that it initially was, right? right? So it moves close together, and the overlap as a muscle contracts, the overlap between thin filaments and thick filaments gets bigger. And a way to easily memorize which filaments decrease in length is the ones that have only one type decrease, because the overlap gets bigger. Whereas something like the A band, which has overlap, it has both, doesn't change in size. Okay. Any questions before I move on after this brief overview? Because I believe, oh, actually here, I wanted to show you this image beforehand. So this is a relaxed muscle, and this is a contracted muscle on the bottom. The blue stuff is the thin filaments, with this red being the thick filaments, right? And everything else is labeled for you. If you notice, the I band here is from here to here, right? Whereas in the final product, it's only like this. So it's got shorter. The H zone initially was from here to here. And then in the end, it's only here to here, right? The whole sarcomere used to be like this. Now it's only like this, got smaller. Whereas the A band used to be from here to here. And now it's still from here to here, right? It's the same length. Just as a heads up, only the blue stuff moved closer in, right? Along with the walls. Okay, so somebody asked during athletics when they have cramps, what is going on? So that's a little bit, we'll cover that later at the end of the lecture, because that actually involves a lot more stuff, but it's basically, it's the same, kind of like the same thing where you have like twitches. It's just over contraction, right? So like if you look at this, the contraction, this is almost complete contraction, because you notice how there's almost no more room for the blue stuff, for the blue thin filaments to go. They're almost max contracted. Whenever you contract a muscle, that's not always the case. For example, just moving my arm like this, it probably is not causing this much contraction. It's causing very small amounts of contraction, because you don't need much more. Whereas if you're lifting, like if I'm lifting like a 200-pound dumbbell and trying to curls with it or something, you would maximally contract, and you would have like to the point where it's actually like painful. Your muscles are pushing, like there's no more room in the middle here for the thin filaments to continue to contract. Okay. So I wanted to ask this question. I'm going to open up a poll. And I just wanted to see if you guys can answer this. Which of the following parts of the sarcomere does not change dimensions, which means just length or size during the course of muscle contraction? So please take some time to answer this. Okay, please take another minute or so to answer this question. And get your answers in. I'm going to end polling. Okay. Okay, so the results, the majority of people picked answer A, let's choice A, the H zone, with some also picking none of the above. So unfortunately, the correct answer is actually B, and nobody's picked it, and I think I know the reason why, right? So when you think of the Z line, you think of the length of the sarcomere, right? That's why you didn't pick Z, because I mentioned that the whole length of the sarcomere shifts. So here, right? You think this is the length of the Z line, and now it's here, and now it's smaller, right? Because these are the Z lines, right? But the Z line, this is not the Z line. This is the sarcomere length. The Z line is this. This is a Z line. This is a Z line. And then in the end, this is a Z line. This is a Z line. So this was a trick question, right? Because look, this length to this length, it didn't change, right? The actual wall itself didn't change length. The distance from this wall to the next wall changed length, but that's not what the answer choice was at. That's not what the answer choice was saying, right? It was just asking for the length of the Z line if it changed, and it does not. Most people pick choice A, which is the H zone. Remember, the H zone is the third zone. So the first zone is thin. The second zone is both. The third zone is the H zone. It's only thick filaments, right? Thick filaments, while thick filaments themselves don't move, this space gets filled up by the thin filaments. Look, in this zone where I drew this line, the thin filaments kind of pushed into it, right? And since the H zone is where only thick filaments are, all you're left with in an H zone in this final is this little tiny piece where there's no blue stuff, right? And you see how much smaller it's got from relaxed state to contracted state, right? Same thing with the I band. Initially start off with there's only this blue stuff, none of this red thick filament, right? And in the end, the only part that doesn't have this thick filament is like right here, right on the edges. So it got much smaller, right? Another component, as I mentioned, that will not change length, but it wasn't an answer choice, but it also would have been a correct answer if it was, is the A zone. The A zone is from here to here, right? And in here, it's from here to here, right? And you can see it's exactly the same length in both. Does that make sense, or do you want me to go over it? So this was a trick question, yeah. And the MCAT will try and trick you like this. Any questions? Okay, if there's no questions, I think we can move on. And feel free to type in the chat if you have any questions or raise your hand or anything.